Alright, so welcome everyone, and now we will be discussing about the cells. Alright, so far we have been talking about the epidermis, right? And in this we have discussed about keratinocytes, Langerhans cells, melanocytes, Merkel cells, right? And now we'll take this opportunity to just talk about in detail about all these things. Okay, so let's just start with the first one. Okay, and I hope you guys remember the location, right, for all the different type of cells. Okay, and we will be covering everything. Fine, so let us start. The first one, that is keratinocytes. Yes, these are the most number of cells are the keratinocytes, right? And these are ectodermal in origin. Okay, that is our first point. Yes, and the non-keratinocytes, these are the Langerhans cells, Merkel cells, and melanocytes. Okay, all right. So this is just like a differentiation, right? So we already know about 4 types of cells. Okay, so first start about keratinocytes. Okay, and in this we will be talking about a very good concept, okay, called as keratinization. Okay, so let us start. Okay, so keratinocytes, yes, they are named after keratin. Yes, and what does this provide? It provides a very good mechanical support. Yes, so let us just start from this mechanical support. So which layer do you think should have a very good mechanical support, and which layer should be like, okay, if it doesn't have, it's okay? So the answer would be, just think about our mnemonic. Yes, which layer was it? Which layer was it? Yes, so it was the stratum basale. Do you think it needs keratin, or the stratum corneum which needs keratin? Yes, alright. So think about which layer needs the most amount of strength. The most amount of strength will be given by keratin, right? Okay, so if any layer needs more strength, so more keratin will be given. Okay, and if a layer doesn't need that much strength, okay, no keratin. Okay, so and keratin provides us strength to the cell. Alright, so let us go on. Start with this. Right here is increase in the strength, increase in keratin. Okay, simple. In the stratum corneum there is maximum keratin, okay, and in the stratum basale there is minimum keratin. Okay, alright, so this is one concept, I wanted to just teach in the first place. Okay, and alright, so they provide us mechanical strength to the cell. Okay, and they divide and generate the layers above. Okay, so fine, the layers above, yes, so it means that from the stratum granulosum and above, yes, they will produce keratin, yeah, to the layers above the stratum granulosum. Okay, so just have that diagram in our head, right? Okay, stratum granulosum, good, right? So it's up, and all the layers above it will have keratin. Okay, alright, so simple, the most easiest definition, yes, the process of increasing keratin in the cells is known as keratinization of cells. We all are happy with the definition, right? Okay, fine. So coming on to the next one, yes, so in the epidermis, yeah, we can just say like this, it's that all of these cells in the epidermis are keratinocytes, yes, and other cells are the immigrants. It's a pretty hot topic on the internet, right? But still, the melanocytes, Merkel cells, and the Langerhans cells, these are immigrants. You can just think this way, okay? It can help us know like which is the most. It's mostly covered by keratinocytes. That is about 90 to 95% of keratinocytes are there. All right. The next thing is, yeah, that is the point, and 90 to 95% cells in epidermis are keratinocytes. The rest are all immigrants. Okay, we can remember like this. Okay, so moving on to the next one. Okay, fine. We have cells, we need some strength. Okay, we added some keratin to it. Okay, what if we wanted more strength? Yes, could we do something? Yes, we can do something. We can add some connections, right? One cell can hold another, one can hold another, and so we can have a good tensile strength, right? So how would we do that? Okay, so we could add something known as filagrants. Okay, does it ring a bell? It definitely does. Yes, so that is the next point. So increasing the strength in keratinocytes by interconnecting with filagrants. Okay, so this opportunity I'll go back to the previous lecture, okay, and I'll show you where did we read it. Okay, so question to you, where did we read it, in which layer did we read it? Okay, fine. Okay, so let's go check. In, yeah, right here, we read it in stratum granulosum, right here. Yes, filagrins. Okay, so in stratum granulosum we are making this from pro to filagrin, and then it was adding some strength. Okay, alright, so it was adding strength by intermediate filaments. Okay, alright, so that was the concept behind it. Alright, fine. So let us go to our topic. Alright, fine. So filagrins, more strength in keratinocytes by interconnecting with filagrins. Okay, fine. So in upper layers, yes, we have filagrins. Yes, so therefore in granular layer the keratohyaline granules contain filagrins. Okay, so that was the point we just saw. Like we went up our notes and we saw in stratum granulosum we have keratohyaline granules, and they make the filagrins, okay, to add tensile strength. Okay, so fine. Now let us move on to the next one. Okay, fine. So this is abdominal keratinization. Okay, so now we know about keratinization is okay, fine. It is adding some strength. Okay, yes, and but if this is not there, or if it is there too much, so what's going to happen? So let us see. The next 2 points are just related to this. So first of all is parakeratosis. Yes, so what happens over here is we read this point before, right? In parakeratosis the turnover rate increases. Yes, the turnover rate increases, and because of this the nucleus is present in the stratum corneum. Alright, so the cell doesn't have time to disintegrate the nucleus. Okay, so this is parakeratosis. We read about this before as well. Okay, so the next one is a very interesting concept, very interesting presentation, is dyskeratosis. Okay, fine, we know that the layers above the stratum granulosum, they're getting all the keratin, right, and the layers above will have filagrins and everything. Okay, fine, we know that much. Okay, but let us see about the normal findings. Yes, so let us have a revision and then we can have the normal conditions as well. So in this condition, what's happening is there is prominent keratinization. Okay, fine, where is it happening? You know, because on the top layers is already we studied about keratinization, we need keratinization, right? But what's actually happening is, if it occurs in the lower dermis, do you think you need keratin? Tell me, do you need? We do not need keratin, right? We do not need so much tensile strength. We need keratin just in the top parts, right? So if there is premature keratinization, right, so what's going to happen? This is a condition called as Darier's disease. Yes, right, Darier's disease. So this is the very basic etiology, right? We know the point of the Darier's disease, that there is abnormal keratinization in the lower dermis itself. Okay, alright, so fine. What findings are we going to see? We will see in the next slide. Okay, alright, so Darier's disease. Yes, so we are going to see abnormal keratinization. Yes, a prominent keratinization. Yes, we'll see in the lower dermis itself. Okay, alright, and these abnormal overkeratinized cells in the lower dermis, yes, there are two types of findings in this. Alright, so these two names we have to remember. I'll just mark them out. Yes, this is corp round and corp grains. Yes, and the next one we'll see them as well. Okay, we'll see the histological image as well. Okay, so what is corp round and what is corp grain? So these are the round shaped cells in the lower epidermis. Yes, and the corp grains are horizontally elongated shaped cells in the upper epidermis. Okay, alright, so this is histological findings. Okay, and Darier's disease, yes, we will be reading more about it as well. Okay, alright, fine. So because of this, what's happening is the cells are not able to adhere to each other. Alright, so whenever in dermatology, and when cells are not able to get attached to each other, the terminology uses acantholysis. So acantholytic cells will be seen. Alright, so cells which are not able to hold on to the surrounding thing are called as acantholytic cells. So these are seen. Okay, and therefore this keratosis is often associated with acantholysis. Okay, This keratosis is often associated with acantholysis. Alright, fine. Cool. So now let us go on to the two things we just mentioned about the corp grains and corp rounds. So grains was horizontally elongated cells and corp rounds were the circular cells we can see over here. This is corp rounds right here, this one, and these are corp grains, these cells, right? These flat cells. Okay, all right, so that is the end for the keratinocyte. Yes, so now let us go on to the next cells. The next cell is a melanocyte. All right, so over here we know we read about melanocyte. Where do we find it? Yes, melanocyte we find in the boys, right? The basal, yeah, stratum basale, we find melanocyte. Okay, all right, so we'll read about this. Where do we find in stratum basale? Yeah, this thing we discussed. What is the origin? Is neural crest origin. Yes, this is also a very good point, right? So neural crest in origin, melanocytes. Okay, and content is melanosomes. Yes, melanocytes produce melanosome produces melanin. We will read about this in a while. All right, so let us continue just with the basic things. Function, production of melanin, okay, and protection from UV light. Okay, and markers, these are important. Yeah, what are the markers for melanocyte? Yes, but why do we need the markers? Yes, if there is a malignancy of melanocyte will cause melanoma, right? Okay, fine. So this will be the diagnosis. To make the diagnosis of melanoma, right, so we will have to memorize these two things, right? This 𝗵𝗵𝗴𝗿𝗺, S100, and melanin A, right? These are the markers for a melanin. Okay, and now let us just see how the melanocyte works. Yes, and there's one good question ahead, so let us see. So in this diagram I'll just yeah, okay, fine. So this is melanocyte, you see, okay, and these we see that over here, yeah, over here that's where the cells are. The cells is making something, okay, and then the cell is transporting something. Okay, this is a regular keratinocyte. The light blue one is a regular keratinocyte. Okay, you get it? It's a regular keratinocyte. Okay, and this is these are the dendritic processes. Okay, all right, and this is a melanocyte. Okay, fine. So the point is that melanocytes contain melanosome, all right, and the melanosome, yes, produces melanin pigment. All right, so let me just write that point real quick. That is, okay, that is a melanocyte. Melanocyte contain melanosome. And the melanosome contain melanin. The reason I have written this at the very top of our page is because it's little bit confusing, so it is better we just remember it as a whole sentence, right? So melanocyte contain melanosome and melanosome makes melanin. All right, fine. So the next question I want to ask you this is right here. So what do you think is the difference between melanocyte and a keratinocyte? All right, so what is the difference? Yes, the answer would be that there are no desmosomes in what? There are no desmosomes in melanocyte. Okay, that is the answer. So there are no desmosomes in melanocyte. So let us just write it down, that no desmosomes. No desmosomes in melanocyte. Okay, alright, so that is the one. Okay, so let us move on to the next question. Okay, I hope you guys didn't saw the bottom part of it. Okay, so color of the skin depends on what? Alright, so $1,000,000 question, right? So color of skin depends on what? So first answer, we think about what is it, right? So we read the question carefully. Yes, the color of the skin does not depend on what? Yes, the question is does not depend on what? So it does not depend on what? Melanocyte. Okay, alright, so as ironic as it sounds, it does not depend. Okay, alright, let me just write it down, melanocyte. Okay, alright, so it does not depend on melanocyte. Okay, so fine, then what does it depend on? Over here we just read something, right? We just learned that melanocyte contain melanosome, melanosome contain melanin pigment. Yes, these things are the one who are responsible. Alright, melanin pigment will give us color. Alright, and the how much amount is being transferred, yes, from the melanocyte to melanosome, and the number of melanosome, yes, that is there in the cell that will impart the color to the skin. Alright, so let us just come to these three statements, important statements. If you don't know the concept, 100% we can fall in a pit and get it wrong. Alright, fine. So amount of melanin inside the melanocyte, number and size of melanosome, and degree of transfer into the melanocytes. Okay, alright, so how much does it transfer, what was the amount and quantity. Okay, so these things are the ones which are responsible for it. Okay, so the next thing we read is a type of melanin pigment. Okay, so as we know, there are a lot of different skin colors, right? But more or less, yes, we just differentiate into two types of melanin, right? That is, first is eumelanin and other is pheomelanin. Okay, and in this one, yes, so pheomelanin is more of yellowish red, more seen in Caucasian people, yes, and the eumelanin is seen in more in black. It has black and brown pigment. Okay, alright, so that was all about melanocyte. So the next cell is Langerhans cell. Alright, so do you guys remember where was the Langerhans cell? Right, so do you guys have that pictographical memory? Yes, we labeled the diagram, we did this, we did that, and everything, Which layer was it? Yes, in which layer is this? Stratum spinosum, right? Smart. Yes, spinosum. Yes, so big smart guys love coffee. Yes, big smart guys love coffee. So yes, spinosum. Alright, fine. So stratum spinosum. Yes, so the origin is a mesodermal origin, bone marrow. Yes, okay, and contents are the Burbeck granules. Yes, we'll see about this in a minute, right? Their contents are Burbeck granules. Function, APCs, antigen presenting cells. So if you have done your pathology right, I hope you have. Yes, so it is APCs. Yes, antigen presenting cells. So what these do is they would take up the antigen, yes, and they'll process it, yes, and they would show it up on their surfaces so that the macrophages can come and process it and create a response, right? So antigen presenting cells, this is a quite an interesting topic. Yes, APCs. Okay, alright, so the next one are the markers, right? So APCs, so why should we do markers for APCs is probably if we can pick up in a malignancy and we can have a positive diagnosis if we find these, right? So the two markers are S100 and CD1A. Alright, so these two are the most important ones, yes, and CD207 marker, alright, for the Langerhans cell. Why should we know it, right? So if there is malignancy, right, if there is malignancy in the Langerhans cells, yes, so it can cause Langerhans cell histiocytosis. Okay, fine, so let us just write that down. Langerhans cell histiocytosis. Okay, so this is a malignant condition, right, or for the Langerhans cells. Okay, so what was for the other guy? The guy we just completed, the melanocyte, right? So melanocyte have some malignancy called as melanoma. Okay, all right, fine. So let us come to the next thing. What are these, right? So these are what? So first of all, I would ask you guys to, how would you describe this image, right? So do you think it's a histological image or taken from a phone, or what do you think? Which image is this, right? So this is from electron microscope, right? So we definitely cannot see this from a regular microscope, right? So these are the 𝗵𝗵𝗵𝗵 granules. Okay, so and we can describe this as a tennis racket appearance. Okay, so this very classical word, tennis racket appearance, 𝗵𝗵𝗵𝗵 granules. Okay, very important. Okay, tennis racket appearance. Yeah, we mentioned here that tennis racket appearance. Okay, all right, fine. So that is for the Langerhans cells. Okay, all right, it has 𝗵𝗵𝗵𝗵 granules and we see a tennis racket appearance. Okay, so now let us move on to the next cell. All right, that is a Merkel cell. Okay, so Merkel cell, where was Merkel cell? Yes, it was at the very base, right? So boys base, right? So what does it do? Yes, it has, so it is for fine touch. Yes, fine touch and 2-point discrimination. Yes, it is a mechanoreceptor. Fine, so let us just write all these points and mind you, all these are very important points. Okay, so the location is Stratum Basale, origin is ectodermal origin. Okay, and content are the neurosecretory cells. Okay, and function, yes, function, it is fine touch. Fine touch and mechanoreceptors. Mechanoreceptors. Yes, okay, and this is a slow adapting receptor. Okay, all right. So what is the marker for this? Yes, as we read for the other two, right? So the marker for this is cytokeratin 20. Okay, cytokeratin 20. Okay, right. So if this has any malignancy, right? If the Merkel cell has any malignancy, the Merkel cell carcinoma. All right, so pretty simple. Merkel cell. All right, fine. So that was all about the Merkel cell. Yes, so all the cells of the epidermis are completed now. Yes, so now what we will do is we will just go on to we'll just connect little bit of physiology in it. Okay, all right, so we just got done with Merkel cells. Yes, and those cells were responsible for fine touch. Yes, and 2-point discrimination, that will come in the list below as well. But now, because we just touched on to the Merkel cells, it's better to just discuss the touch receptors. Yes, all the mechanoreceptors. Let us quickly revise them. Even though this topic is from physiology, right? But this is still one of 19, right? So we should know it. Fine. So let us get this opportunity to just revise these things. Okay, so Merkel cells we just got completed, right? Meisner's corpuscle, Ruffini corpuscle, and Pacinian corpuscle. Okay, so Pacinian corpuscle is very famous for this structure, right? This one is Pacinian corpuscle, and this is a Ruffini corpuscle, and this is Meisner's corpuscle. All right, so let us go and discuss each of these. Yes, and we'll see how to remember them. That is the most important one, how to remember them. All right, so there are specific things, we just follow them and we'll never go wrong. Okay, so Meisner's corpuscle is for gentle tap. Yes, gentle tap, low frequency vibrations. Okay, low frequency vibrations we have Meissner's corpuscle. Okay, so how can we remember this is just with this one sentence. That this is how I remember it, like Miss likes fine touch. Yes, so Miss likes gentle touch or Miss likes fine touch. Yes, so we can remember this, and we'll never go wrong with the Meissner's corpuscle, right? So Miss likes gentle touch. Okay, where do we see it? On non-hairy skin. Okay, so it's going to be on the lips, palms, yes, and non-hairy skin we'll find these receptors. Okay, all right, so low frequency vibrations, Meissner's corpuscle. Okay, fine. The next one is the Merkel cell. This one we read a lot about, right? So but how do we memorize it? That is very important. So this is exactly how I did it. So I'll be writing a sentence here. So Merkel, we just remember, right? Merkel seems like a name. Yes, Merkel seems like a name of somebody, right? So I remember like Merkel is name of blind boy. All right, so if you remember this sentence, you will never go wrong, I bet you. Okay, so Merkel is the name of a blind boy, and blind people use braille for reading, right? Yes, so because of this, so to read the braille script we will need 2-point differentiation. Definitely we will need, right? So because this script is involving these sort of elevations on a hard surface, right? Yes, so 2-point differentiation is by Merkel cell. So this is what it's written here. Yes, so yeah, best in 2-point discrimination, helps in reading braille script. Okay, so that is very important. Okay, so just keep this sentence in the mind and you will never go wrong. Okay, so Merkel is a name of a blind boy. Okay, and Meissner's corpuscle, how do we remember it? Miss likes gentle touch. Okay, so now we go on to the next one, that is Pacinian corpuscle. All right, so Pacinian corpuscle, these are the ones who are like very deeply placed. Okay, and yes, these concentric ones, yes, these are Pacinian corpuscle, like, so because they are very deep. Yes, so these are being used for high frequency vibrations. Okay, yeah, high frequency vibrations, yes, and we are the most sensitive ones for the high frequency vibrations. Okay, all right, more sensitive for high frequency vibrations. Okay, and crude touch, deep touch. Okay, because they are deep, they are crude. Yes, crude touch. Yes, okay, fine. So we just have to remember it just like this, that so Pacinian corpuscle, like, so we just have to remember this, right? So okay, fine, this is concentric looking cells in the deepest layer, used for crude touch. Yes, that is very important. Okay, so 3 things we remember in this one. Okay, all right, so now let us go on to the last one. Yes, that is Ruffinius corpuscle. Okay, so where do we use it? Yes, so this is for the skin stretch. Okay, this is for the skin stretch, and how do we remember this? Is whenever we wear a jeans, yes, and sometimes it gets a little bit maybe pulled, right? It gets pulled for some reason or the another, right? So it can have a little bit of that shredding, right? Little bit of shredding. So usually what we do is when this thing patches, when there is a patch, right? So we would just get it removed. Yes, so just to get those 2 things together. Yes, so that is where this thing comes from. For Ruffinian corpuscle, we have skin stretching. Yes, so skin stretching is being picked up by Ruffinian corpuscle. Alright, so that was all about the touch receptor. So let us just revise this once again. Alright, so and just to give this a good conclusion. Yes, so first of all we have the Meissner's corpuscle. Yes, for gentle tap. Yes, and how do we remember it? That Miss likes gentle touch. Yes, so Miss likes gentle touch. This is how we remember. Okay, how about the next one is the Merkel cell. Merkel is the name of a blind boy used for 2-point discrimination. okay, all right, that is for Merkel cell, okay. And the third one is the Psoriasis corpuscle. Yes, this one was for the deepest layer, right, and there were those concentric cells, right, and in the deepest layer, that is why they have crude touch. Yes, crude touch, fine. And high frequency can be, is more sensitive for high frequency, all right. And the last one is the Ruffinian corpuscle, is very sensitive for skin stretch, okay, all right. So that was all for touch receptors. I hope this was a very good, just as a revision, quick revision, right, for all the touch receptors, okay. And I hope the discussion was good and very productive. Thank you so much for listening.